The next speaker will be Dr. Bikbova. She used to be an ophthalmologist in Ufa Eye Institute, and she came to Chiba University as a postgraduate student supported by the Japanese government scholarship. And she finished the postgraduate student school last year, and then now she was appointed as a research scientist in Chiba University.
Добрый день. So, good afternoon, everyone. Let me present our research on drug development for vision loss prevention in diseases associated with ages accumulation. So first of all, what is advanced glycation end products? This is the group of molecules which are forming from non-enzymatic reaction by reducing sugars. And we do accumulate advanced glycation end products by aging, but in patients with diabetes, the concentration of ages is much higher. So this is the reaction of sugars with proteins, lipids, and nucleic acids. So in general, in our body, the advanced glycation end products, they are forming through different stages, and in the early stage, they are by Schaeffer rearrangement, the different, different substrates forming, which in the advanced stage, they are causing the inactivation of enzymes and denaturation of proteins. So as Dr. Ashtari presented before, the mitochondrial caspase-dependent self-death pathway was found to be associated with the neuronal cell death in patients with diabetes. But at least 40% of neuronal self-death in damaged neurites, they should be related to caspase-independent self-death pathway. Also, as he presented before, the neurotrophin 4 has been shown to have the best neuroprotective and regenerative effect under high glucose condition. So apoptosis-induced factor is the first mitochondrial protein which known to work independently of caspase. So, and specificity protein 1, it's the transcription factor which regulates different processes in the cell, such as apoptosis differentiation or immune responses. So for our study, we use the three-dimensional tissue culture where the retina is isolated, the retinal explants are prepared, and they are fixed in a collagen gel and cultivated in the serum-free media as a control. And for our study, we use the very low dose of advanced glycation end products in concentration 10 milligram per milliliter. And to study neuroprotective and regenerative effect of neurotrophin 4, we used neurotrophin 4 in concentration 100 nanogram per milliliter. So here is the regenerated neurons. The pointer doesn't work. So the normal one presented in the above, and you can see the length of it with a growth cone, and under the advanced glycation end products exposure, the number of neurites were decreased, and as you see, they are much thinner and weaker. And addition of neurotrophin 4 increased the number of regenerated neurites and increased the number of them as well. So here you can see that the number of regenerated neurites under the high glucose condition is significantly reduced, and neurotrophin 4 increased the number of regenerated neurites more than 3 times, even comparing to the normal group. So here is a representation microphotographs of tunnel staining, and the blue signal is the ganglion cell layer, and the green signal is the tunnel positive cells. So as you see, the advanced glycation end products exposure increased the number of tunnel positive cells, and neurotrophin 4 decreased that number. So here is the ratio of tunnel positive cells represented. As you see, the neurotrophin 4 decreased the number of tunnel positive cells in all group of advanced glycation end products exposed retinas. So caspase-9 immunostaining showed that the caspase-9 immunopositive cells were increased in the ages-exposed retina, and neurotrophin-4 decreased the number of immunopositive cells. So same findings we observed in the apoptosis-induced factor immunostain group. And to find if there is any biochemical link between the caspase-9 or apoptosis-induced factor, we use the caspase-9 inhibitor, and which shows, so if you see on the top, if you see on the top, so the caspase-9 inhibitor significantly reduced the number of caspase-9 immunopositive cells on the C picture. And on F picture, there is shown that it didn't affect the number of apoptosis-induced factor immunopositive cells in the ganglion cell layer. And thus, this result indicates that there is no biochemical link between caspase-9 and apoptosis-induced factor activation in ages-exposed retina. So thus, we found that both caspase-9 dependent and caspase-9 independent self-death pathways are involved into the neuronal self-death in ages-exposed retinas. So our second experimental design was to study the high dose of advanced glycation end products, where we used 100 milligram per liter of advanced glycation end products. And we studied different neurotrophic factors, such as neurotrophin-4, hepatocyte growth factor, glial-derived neurotrophic factor, and tau-urso desoxycholic acid. So the study had been performed in the same manner. And here is the representation of tunnel positivity. So it was found that the high concentration of advanced glycation end products also increased significantly the number of tunnel positive cells, and all factors decreased the number of tunnel positive cells, but the most significant decrease we observed in the group of neurotrophin-4. So we started the SP1 expression, and we found that all neurotrophic factors increased, the SP1 immunopositivity were increased in the groups of all neurotrophic factors, and it was still remaining high in advanced glycation end products exposed retinas group. So thus we can conclude that maybe SP1 expression works in the regulation for neuroprotection and neuroregeneration. So NF-kappa-B immunopositive cells were found also significantly higher in the group of advanced glycation end products exposed retinas. And all factors decreased that number, but the most significant decrease were observed in the group of neurotrophin-4. So thus, both caspase-dependent and caspase-independent self-death pathway are involved as in a low dose as well as in a high dose ages-exposed retinas. And we found that the expression of transcription factor, NF-kappa-B and SP1 in ages-exposed retina confirmed that the receptor of advanced glycation end products are expressed in the neuronal cells exposed in advanced glycation end products. So this is the scheme, concluding our study. So as you see that both caspase-dependent and caspase-independent self-death pathway involved into the apoptosis and overexpression of SP1 and NF-kappa-B, they confirmed that there is an enhanced interaction between ages and receptor for ages, which causing further pathological changes in the neurons which are exposed to ages. So that's our purpose, was to determine whether the combination of different therapeutic agents will have the greater neuroprotective abilities in the cultured retinal cells exposed to advanced glycation end products. And to accomplish this, we used the same retinal culture system, and we applied several combinations of neuroprotective agents to the culture retina. So the citicoline was chosen as the first therapeutical agent because it was found as a mitochondrial stabilizer, which is working for prevention of the activation of phospholipase A2. And it was shown as a potential therapeutic agent for different conditions such as brain ischemia, Alzheimer's disease, and Parkinson's disease. So the second agent was chosen to be tauroursodesoxycholic acid, which has been found to work as anti-endoplasmic reticulum stress, and several papers they were published to show that the tauroursodesoxycholic acid was shown to modulate the endoplasmic reticulum function to protect cells against this endoplasmic reticulum stress-induced apoptosis. And considering those features, TUDCA was also selected to be the compound of our multi-compound therapeutical agent. So for our third experimental design, we used the high concentration of agents in concentration 100 milligram per milliliter, and we used the mentioned neurotrophic factors: ceticolin alone, tauroursodesoxycholic acid alone, neurotrophin 4, and their combinations: the duplet, which consists of ceticolin and TUDCA, and the triplet: ceticolin, TUDCA, and neurotrophin 4. So the study had been performed in the same manner, and this is the result of neurite regeneration. And for control, we also used the AGES inhibitor. So as you can see there, unfortunately I cannot point. So the neurotrophin for the duplet and triplet, they shown to have the best neuroprotective effect. So the most significant effect, of course, we observed in the group of triplet. So there is the pictures of neurite regeneration. So A is the control serum-free media, G picture is the AGES exposed retina. So, and the most interesting for you will be the E picture, which represents the triplet, and the F is neurotrophin 4, and the D is duplet. So as you see, by the way, I need to mention that we used in this experiment, we used the neurotrophin 4 in a concentration 10 times less than in the previous studies. But as you see here, the neurites, the regeneration of neurites were increased significantly. So, considering the tunnel positivity, it was also found that all factors decreased the number of tunnel positive cells, but the most significant decrease was observed in a group of neurotrophin 4 triplet and duplet. So in conclusion, the solution containing 3 neuroprotective agents was the best in enhancing the neurite regeneration, more than ceticolin and TUDCA and neurotrophin 4 alone. And this result indicates that the combination of neurotrophic factors may have the potential as axoprotectants for the eye with the diabetic retinopathy. So the duplet and triplet significantly increased the number of regenerated numbers and decreased the number of tunnel positive cells, and thus they should be considered as the possible agents for neuroprotective and regeneration therapy for diabetic retinopathy. And further, we are planning to perform the in vivo study to determine the effect of combined therapy within eye drops in a diabetic animal models. So thank you for your attention.